Okay, I don't enjoy it. Like,
All right. I'm recording now. There's a couple go out in first grade too, so thanks, guys.
Wait, just a second.
No, yeah.
Okay, so is the melanin in your eyes and your skin, are they two different things?
The eye color, yes. But it's sort of like yes, they're different genes.
Okay, so that's why people with really light skin can have really dark eyes.
Yeah, totally, yeah.
is there a benefit for different colored eyes?
Having darker eyes is the same as having the darker color skin. It protects your eyes from UV radiation. Like people that are albino, they have vision problems. Not only because they're really susceptible, but it also helps to like dissipate excess light from getting in your eye, and they just, you know what I'm saying?
All right.
So yeah, there's a benefit. All right, so gene expression, we've talked about this before. Gene expression is the process by which your genotype is basically transformed into phenotype. Gene expression includes DNA transcription and translation. Gene expression, so your gene is expressed or produces eventually the phenotype. Okay. The beginning of your chapter, there's that side page where it talks about an experiment that was done. Some of you might have skipped over it when you read or took notes. But by studying metabolism is the breakdown of stuff. Okay, so when you metabolize your food that you eat from your lunch, you break it down into component monomers and you can absorb it in your large intestine. But sometimes metabolism also indicates just different chemical reactions. So in your metabolism, part of what your body does is synthesize things like amino acids. And a lot of times it doesn't just happen in one fell swoop. You turn X into amino acid tyrosine. It happens in many different steps. You make precursor and change it to something else, and eventually it is arginine or lysine or whatever. So a lot of times there's different steps in metabolism that are each catalyzed by a specific enzyme. So for example, if you start with your precursor A and you want to convert it into the amino acid, let's see, tyrosine E. Well, there's a lot of different steps in the middle. You don't just go from your precursor all the way to tyrosine. So anyways, by physicians or people that study metabolic defects, you can learn a lot about metabolism and about enzymes. There's a couple of different disorders that are caused by defects or just your genes, mutations, and different genes that code for enzymes and the production of, or the breakdown, of phenylalanine. And so phenylalanine, we talked about PKU before, right? Phenylketonuria. Not talking about like a Diet Coke or something. Did I not miss that?
No.
Oh, damn class.
Wait, Miss Sandra, what happened to your bottle up here?
Put them up there.
That's so long ago.
I know.
All right, I'll pass around my Diet Coke first, and then I'll sanitize it. But if you notice on here, I don't want blue, right? Look on the ingredients, and you'll notice on here it says phenylketonuria contains phenylalanine. On almost any diet drink, Diet Coke, Diet Pepsi, Diet Dr. Pepper, whatever, you're going to see that on there. Phenylalanine is an amino acid, and we have the enzymes to break it down in this particular pathway. Some people that have a genetic mutation in such a way they can't break down phenylalanine, they have a disorder called phenylketonuria. And this enzyme that breaks down phenylalanine is blocked. It doesn't work. And so they produce phenylpyruvic acid, and that can build up in their body and can cause problems in mental development. And so newborn testing, when newborns are born like in a hospital, the babies are tested for specific genetic disorders right away, with not even parent consent, like it just happens. One of the things that they're tested for is PKU. And if you found that your child has it, you just avoid giving them stuff with phenylalanine in their diet, and they're completely fine. So there's really no treatment for it. You can't cure it. You just avoid phenylalanine, so they can't drink a lot of diet stuff or whatever.
Are there a lot of things that you guys have it?
I don't think. It's in the synthetic, like, it's in the artificial sweetener is why it's in the diet drink. I don't think it's, like, super common. And there's another one, alkaptonuria. They pee black. Their
That would
urine
be
looks
so
black. That would be fruity.
That would be cool.
The alkaptonuria way down here, the last enzyme in this pathway is not defective. It doesn't work. Anyway, so moving on. There's an experiment conducted by these guys, Beadle and Tatum. There they are. It was on the cover of Time magazine in like 1949 or something. Anyway, a long time ago. But they were interested and they had an idea that each gene codes for one enzyme. Every gene in our body codes for one enzyme. That was their hypothesis. So they developed an experiment to test their hypothesis. And I'm just going to kind of go over to a picture and describe it. This is all in your book, like I said. And they're working with a fungus called Neurospora. It's a fungus. Like, think about, like, you ever in elementary school, you leave your bread out for a long time and like you're seeing a house staff of gold with girls or something. Yeah. Like, that's a fungus. This little fungus, Neurospora, is like a bread mold type of thing. Are we done? Keep looking at sun. Oh, very good.
Oh, right there. It's in bold letters.
Anyway, they understood that these scientists understood enough about a particular pathway in Neurospora that they could synthesize arginine. Arginine is one of the 20 amino acids. And Beadle and Tatum knew that if these fungi were given what we call minimal medium. The medium is like agar. You can grow bacteria on agar, you can grow fungus on agar. Okay, a lot of times when you grow bacteria, you can have contamination and fungal contamination. If you have this regular minimal agar with minimal nutrients, with this precursor in it, normal or wild type Neurospora fungus could convert it all the way to arginine. They understood enough about this pathway. They knew that there were different steps, and that the different steps included a precursor, was changed to something called ornithine, was changed to something called citrulline, which was eventually converted to arginine. They understood this whole pathway. They didn't understand that, like, were these enzymes coded for by one gene or not? They didn't. That's what they were trying to test. So, and they knew that each step was catalyzed by a different enzyme. So they called an enzyme A, enzyme B, and enzyme C. And their hypothesis was these was catalyzed, coded for by different genes. So gene A, gene B, and gene C. The scientists also knew enough about radiation to know if you expose, not that you're sick, but stuff to radiation, it will induce mutation. What they did was they developed 4 types of agar, basically. This one they called minimal medium. It had basically almost nothing, just minimal nutrition. And the other types, there were 3 other types, were supplemented with the different steps in the pathway. So this medium contained ornithine. This medium, the third one, contained citrulline, and then the last medium just contained arginine. Such that if what they did was they exposed the fungus to X-rays, like large doses of X-rays, to induce mutation. So if a mutation was caused in the first enzyme in the pathway, this right here, they would say, okay, if you're given the precursor and they have this mutated here, would it be able to grow if it was just given this precursor? So the fungus can't grow without arginine. It has to be able to make it, otherwise it can't grow. So if you grow it on this minimal medium with just the precursor, but it induced a mutation here, technically it should not be able to grow because it wouldn't be able to convert it to ornithine and it couldn't go all the way. Does that make sense? But if it was that same one, if it was given ornithine in the media, it should be able to grow. And then obviously it's given the other ones because these enzymes should still be okay. They also thought that, okay, so if you induce a mutation in the second gene, then it shouldn't be able to grow in either one of these mediums because it could maybe convert this to this and it would be stopped here. Couldn't convert ornithine into citrulline and therefore wouldn't grow in either one of those. And if the mutation was induced in gene C, the last enzyme in the pathway, it shouldn't be able to grow in any of these media because the last step would not be able to happen. Does that make sense? So they exposed the fungus to radiation and they grew it on all types of agar. Now they didn't know which was going to have which mutation, but they observed what agars it grew on and they classified them based on what medium they could grow on. So they called the class 1 mutants, the ones that were able to grow on all three except for the minimal medium. And so they said, all right, so these guys must have a mutation in that first gene, gene A. The ones that could grow on the last two but not the first two, they called class 2 mutants. They induced a mutation in gene B. And then the last were class 3 mutants. Anyway, this supported their idea of one gene, one enzyme. That's what it makes sense. Reading your book. The only way you'll get this is if you'll given, like I said before, the experiment and the results, and you have to interpret it. You don't have to memorize it. Okay, so one gene, one enzyme. There's a problem with that hypothesis, however, and it's that not all proteins are enzymes, but all proteins are coded for by genes. So then what could we say? It's not that, then it's one gene, one protein. Great, okay. There's a problem with that. In that, a lot of proteins are made up of several polypeptides, like hemoglobin is made out of 4, collagen is made out of 3. Most of our functional proteins are made up of multiple polypeptides, and a lot of times each polypeptide has its own gene. So then you can say one gene, one polypeptide. But there's a problem with that. And that a lot of genes only code for RNA, ribosome RNA, transfer RNA, silencing RNA, there's different types of RNA. So then you can't say one gene, one polypeptide, or one gene, I don't know. You could say one gene, one product. Sure. But there's a problem with that. And that a lot of genes code for more than one product. Some genes can code for multiple polypeptides. Some genes can code for RNA or a polypeptide. It's hard. So in 2003, this lady kind of wrote a paper and said defining a gene is problematic because one gene can code for several protein products, like down here. One gene or some genes can only code for RNA. 2 genes can overlap, and there's a lot of other complications. So defining a gene is not so easy. But we need to understand the process of going from DNA to RNA, or going from DNA eventually to polypeptide. So we understand how genes are expressed. So gene expression doesn't necessarily have to be a protein product per se. Genes can be transcribed just to make RNA. Anyway, just to give you an idea, I've mentioned this before about how science changes so dramatically over time. 2003, I was already teaching. I had just graduated from getting my master's degree in grad school. But when I was in high school, like, this hasn't even been published yet. They didn't really know. So a lot has changed in just, since I graduated high school, and a lot has changed since then. So keep that in mind. It's interesting over time as science develops and they learn new things. You can compare to what you learned like in high school or whatever. And anyway. Yeah. In turn, they understand a whole lot more about gene regulation and what the knowledge of what we thought was junk DNA is actually junk. We'll talk about that in another time.
I get all my facts from Twitter.
Huh?
I get all my facts from Twitter.
Just like make sure you're conscientious of the sources.
Most of them.
Oh, well, okay. Those are good. The scientific one, like science, yes. In chapter 21, we'll talk about a very new event, which you might have seen as the cloning of a monkey.
I saw that.
Chapter 21 is our biotechnology chapter. We'll talk about that.
Did you retweet stuff about that? I know I saw it on my timeline. And so, they were like, you know what? They might do it on
Unethical, but yes.
That's so.
Unethical, but yes. We'll talk about humans and all that stuff later.
Women clone all the time when they have identical.
But that's not a clone of the mom.
Oh, yeah.
And you can't really help that from having to raise them. All right. Anyways, so transcription to translation. DNA to RNA is transcription. RNA to protein is translation. That's what we call the central dogma. Anyway, so that's what we're going to talk about in this chapter, is how all this works. So first of all, if you are a bacterial cell, where are your genes? Floating around in the cytoplasm. Yes?
Prokaryotes do not have organelles.
Right, that was me. If you are a eukaryote, like in this picture, your genes are in your nucleus. Yes? Okay. Make sure we delineate. You have to constantly think about the differences. I'll keep bringing them up, but just keep that in mind. where protein's made no matter what you are, bacteria or eukaryote. Ribosomes, which are located where?
On the cytoplasm.
In the cytosol. They could be, but they're in the cytosol.
Oh.
They're not in the nucleus, is what I'm trying to say. They're in the cytosol. There you go. Good. Okay. So if you are a eukaryote, how does the information get to the ribosome? Does the DNA leave the nucleus? It's RNA. So DNA can't leave the nucleus. The DNA is sort of like, it has the instructions for how to build polypeptides. Sort of like an architect has the idea for how to build a house in his head. But if you're my builder, I can't like just be like, take my information. Like, it's not going to just be transferred to you. I have to draw it, write a picture, do a bunch of stuff, and then give you the blueprint, and then you build it, right? That's the same thing here. So the DNA contains the instructions. They have to be transferred to a movable form, which is the RNA, messenger RNA. That will move to the ribosome, and then the ribosome will read that and produce the polypeptide. Okay? So the DNA does not leave the nucleus. So the messenger RNA is what carries the information on how to make a polypeptide. Let's talk about RNA for a minute. RNA is a polynucleotide. So it's made out of nucleotides that held together with phosphodiester bonds. It is only one polynucleotide. It's not two wrapped around each other like DNA. It's just single-stranded, usually. Now, RNA can be double-stranded in some instances. It can base pair with itself. I can transfer RNA, or in the SNRPs for the spliceosomes, which we'll talk about later. I know it's a fun way for it to say SNRPs. But in general, we say single-stranded. The bases in here are adenine, guanine, cytosine, but instead of thymine, there's uracil. Okay, so if you're going to base pair here, you're going to do A to U and C to G. There's lots of different types of RNA. There's messenger RNA, which is the information on how to make polypeptides.
It's
a big brownie.
It's bigger at home, of course.
Oh, did you like?
I get it from work.
Oh my gosh. You know, I'm sorry. Please. I'm hungry. Publix cake is like my favorite cake in the world. Oh, like Publix, I know. Mary Frances last time was like, Mommy, what kind of cake do you want for your birthday? Let's make one. I was like, no, Publix cake. Tell Daddy Publix cake. Anyway.
Oh, it's coming up though, isn't it?
March 8th.
That's close, March 8th. There you go. What is yours? Oh, February 8th? This is on February 5th.
Eddie's anniversary. Oh, that's our.
Oh, someone's birthday is on the 5th. That's right, but y'all say, yeah. Nathan, that's right. My dad's birthday. Anyway.
What is that? Have you seen the movie Dad?
Yeah.
Can we watch it?
After the AP exam, yeah.
Okay.
I would show them my bio 1 kids like during the semester, but we got to get through. You don't even know how much we have to do. We're not as behind as Richie though. I was talking to that teacher on Friday. He was at the thing at JFHS, and I was talking to him and like,
What section are they on? What section are they on?
They're doing like chapter 13 through 15 right now. Okay. And he hasn't done any organ systems or anything. We've done at least a few. Anyway. So messenger RNA is for building polypeptides. Ribosomal RNA, rRNA, is what helps to make up ribosomes. tRNA is transfer RNA. That brings amino acids to the ribosome during translation. There's siRNA, which is silencing RNA. That's a regulatory molecule. And there's all the other types that we'll talk about a few as we go on through the rest of the year.
What is RNA?
Ribosomal RNA. RNA is multifunctional. It can have a lot of functions, and it also can serve as an enzyme. RNA has enzymatic capabilities. We call RNA that can function as an enzyme a ribozyme, by the way.
Oh, that's what that was. Yeah, from the notes, like.
Ribozyme.
Okay.
What's interesting is that in terms of like evolution, the genetic material that is most likely to have evolved first is RNA and not DNA, because RNA is single-stranded, it's a little bit simpler in structure, and it also is multifunctional, and it has enzymatic capabilities. So they think that RNA evolved first, and the DNA evolved because it was a little more stable. So anyway, we'll talk about that when we do our evolution stuff. RNA that has enzymatic utility.
That's like just evolution.
Like I said, it can base pair with itself, like in tRNA. It can fold on itself. Yeah. Well, sort of. Like, so the siRNA, I'm going to answer this to you and not the whole class. When, like, an mRNA is made in the nucleus, there's different levels of what we call gene regulation. So preventing gene expression, there's all kinds of ways that can help. So if a gene is transcribed and makes an mRNA, you don't want the gene to be expressed, you don't want to fit information like that. The cell can make silencing RNA molecules, silencing RNA molecules that will here to the RNA, the mRNA in the nucleus, and that signals that it can be created for it. So it can make it.
But why would the siRNA want to do that?
If the cell needs to prevent a gene from being expressed, but it's sort of caught it too late, like after it's already been transcribed, but you want to prevent it from being translated, you kill the RNA basically before it leaves the nucleus, you in fact silence the gene.
So, like, if it was a skin cell or something that tried to make a protein that a skin cell shouldn't, it would send that out to stop it?
Yeah.
Okay.
Yeah. So without it's chapter 18. Yeah.
Slightly off topic. What's your favorite public school?
Oh, I like just the regular sheet cake, the chocolate or chocolate vanilla. Not the fancy ones. They make all kinds of fancy ones. I just like regular sheet cake. The marbles,
That's
all right, ready?
Yeah. DNA, RNA,
Oh, I like that. You got the writer, I think like. Although she likes like Walmart, she likes, she's not.
I like Fonda.
Oh, why?
It's not good.
I like the way it feels. It tastes like
Yeah, like you had a great
All right, let's talk about transcription. There are 3 stages. Now, transcription is going from DNA to RNA. There are 3 stages: initiation, elongation, and termination. We're going to talk about the different stages, and I think this slide is, well, it doesn't cover anything yet. The first, I'm going to talk about bacteria first, and then I'll come back and say a few differences in eukaryotes are. So right now, we're kind of talking about bacteria. In general, so there's only one enzyme involved in transcription. It's called RNA polymerase. The only one. This big pink thing right here. RNA will come in to a specific part of the DNA, the gene that needs to be transcribed. It will unravel the DNA around 20 to 30 base pairs at a time, and it will read one of the strands. The strand that it reads is called the template strand. That's the strand that's transcribed. The other strand that's not transcribed is called the coding strand. It's just what it's called. It will read and build an RNA molecule complementary to the template strand. So if it finds an A, it will add a U. If it finds a T, it will add an A. On and on and on and on. These nucleotides come in with 3 phosphate groups, so they bring their own energy. The RNA polymerase will catalyze those phosphodiester bonds being produced. As the RNA is being synthesized, the RNA polymerase will move on. The RNA just kind of pokes out of the enzyme and just will stick out. And when it's finished, the RNA will be released. The DNA, once it's passed over, once the RNA polymerase is passed over the region it's reading, the DNA will just coil right back together. That's generally how it works.
You have a video from yesterday lesson?
I will show you a video when I'm finished talking about all of it. This wasn't all of it. I'm talking about the stages now.
Do they know exactly why it uses uracil instead of uracil? Sorry.
Sorry, Katie. We don't make
There's an answer somewhere. I don't know what it is off the top of my head.
Okay.
Initiation is the first stage. Stage 1, initiation. Remember this right now in particular is bacteria, prokaryotes. So what happens is that the RNA polymerase will bind to a region on the DNA called the promoter region. The promoter region is the specific sequence of DNA nucleotides that signals the RNA polymerase, hey, bind here. That region is not transcribed, it just signals where the RNA polymerase should bind. It flanks or is at a butt the end of the gene.
You know how like when you were talking about DNA thing, how like it has a hang off or something like that?
At the very end.
RNA group? Oh no, it doesn't. Never mind. It's only single. Yeah. Never mind.
Okay.
Yeah. Thanks.
Okay. So the promoter does a couple of things. One, it tells obviously where it should bind to. So two, it orients the RNA polymerase in the proper orientation, such that it tells it which strand of the DNA to read. Which one is the template strand, which one is the coding strand. The the RNA polymerase will always read the DNA in the 3 to 5 direction. RNA polymerase is like DNA polymerase in that it can only build 5 to 3. It always has to add a phosphate to a hydroxyl. Now it can start with nothing, it can add a first one there, but then from there on it always has to add 2 or 3 times hydroxyl group. So it always builds a 5 to 3, but it reads the DNA 3 to 5. That's initiation. Easy enough. Elongation is where it basically moves around the length of the gene, in this case it's the dark blue, and it will build the RNA transcript or molecule. So it'll just read the DNA, and where there's an A, it'll put a U. Where there's a U or T, it'll put an A. Where there's a B, it'll put a T. Whatever. It builds it complementary. RNA polymerase can make a lot of errors. There's no proofreading ability. So there's one error per about 10 to the 5th bases. It's not a problem though, because a lot of times if a gene needs to be transcribed, it's going to be transcribed many, many times. It can produce lots, like hundreds of RNA molecules. And if some of those RNA molecules have problems, well, there's probably other ones that are fine, and at the end, you'll still produce plenty of functional protein. So it's not a big deal. Also, the RNA molecules that are produced are very short-lived. They don't last for very long in the cytosol. They are degraded relatively quickly. There's a reason to that. It is very important that you don't transcribe random genes at any given time, because there are certain genes in your body that are, like, for example, never turned on. Your skin cells are very different from your heart cells. And the reason they're very different is because different DNA is expressed in those different areas. You don't want to turn on the same genes in your skin cells that are on in your heart. So those genes are turned off. It's also really important in your pancreas, you only actually produce insulin when you have a lot of sugar floating around in your blood. You don't want to just be producing insulin all the time. Insulin is a protein product, so it has to be produced through transcription and translation. So you only want to transcribe the insulin gene when it's necessary. Does that make sense? So it's important that it's regulated. So one of the things that's regulated, well, if you eat once or you eat your nut butter, it's still full of sugar. So right now he's got sugar in it. So pretty soon he's going to start producing insulin to get rid of that sugar in his body. But before that, he doesn't just want random levels of high insulin all over the place. Eventually you'll have diabetes. So those mRNAs, that after you're finished absorbing all that sugar, those messenger RNAs need to be degraded. So he stops producing that insulin. Does that make sense? So it's important that they don't live for very long. And that's really a lie. But, they don't last for very long in the cell. If the cell needs more, well, it's just going to transcribe more of the genes. Does that make sense? Okay. Just a picture. Now, if you are a bacteria, this is happening in your cytosol, in your nucleoid region. That's what's going on. Yay. Okay. Termination is the end of transcription, the end of the gene. If this red area, there's a termination sequence. As the RNA polymerase will transcribe, it actually transcribes the termination sequence. And when it does that, it builds RNA. Well, most of the time, a lot of the time, what it does is it actually transcribes a sequence that causes the RNA at the end to base pair on itself, like this, and produce something called a GC hairpin turn. Notice the letters in here, GC, 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 GC. And it produces like a turn, looks like a hairpin. And it causes the RNA to stick up in the way of the RNA polymerase and the DNA, and it kicks it off. Physically, like busts the RNA polymerase off the DNA, and then transcription is finished. PT your intern. I'm so tired. When my husband gets stressed, he wakes up, and then he wakes me up. Well, so he woke up at like 2 and like went to eat, and then I like wake up and I'm like, crap, I have to pee. And so I peed and can lay back down, and usually I'm like, I don't say a word, I just lay like, I'm like, please don't talk to me. Please don't talk to me. So I can go back to sleep. You know, if I was a nice wife, I'd be like, are you doing okay? And like I did. But then he was like, are you awake? And I'm like, yes. And he's all stressed, and then he talks to me for like an hour and a half. So then it's like 4 o'clock in the morning. I don't go back to sleep until like 5:15. I'm really,
That's what I'm saying.
This is what happens when you get married. I'm sorry. Oh, anyway. Just saying.
That's what
I'm
I'm
sorry.
saying.
Yeah, I mean,
You gotta get it out while it's on your heart. While it's blowing around in your head, you gotta get it out.
yeah, I mean, I get it. It's fine. I was happy to help him, but I'm really tired. I mean,
Is he better now?
yeah.
Imagine what it would
yeah.
have been if
Well,
you
he's got a
were.
lot going on right now. When he gets stressed, like, he'll leave like a 7-minute voicemail on someone's like phone, and I'll leave like a 30-second one. He just talks. Like, he just talks. So when he's worried, he has to talk. Like, when I'm worried, I might say something for like a minute, and I'm like, I'm done. We're just very different.
Talk. You have to talk. Oh my God.
Time is down anymore, and I'm just tired. All right. That's all of transcription. That's bacteria. Eukaryotes is a little bit different, but only in the initiation phase of it. So I'm going to talk about this, and then I'll show you the animation you wanted, okay? Before we get to that, I need to talk about a few differences in the DNA. I've mentioned a lot of this already, but I want to kind of go over it again and mention something I haven't talked about. So first of all, bacterial DNA is in the cytosol and the nuclear region. Eukaryotic DNA is a nucleus. It's more protected there, whatever. Prokaryotic DNA is a circle, and there's only one of them. Eukaryotes have linear chromosomes. There's lots of them usually. And because there's so many, in order for them to fit in that nucleus, they have to be tightly packed. And one of the ways they're tightly packed is to be wound around proteins called histones. Because there's not a lot of DNA in bacteria, they don't have to be tightly packed. They're just hanging out. So we call the DNA in bacteria naked. What that means is there are no histone proteins. It's just hanging out there. I've already sort of mentioned all that before today. The last difference that is really, really important is that there's no junk DNA in bacteria. So introns are what we call junk DNA. Junk or non-coding.
Is that the ones that are super like tight?
Hmm?
Is that the ones that are like super tight, like they're tightly wound so that they can't be read?
No, that would be an entire chromosome,
Okay.
like a bar body. No, what I mean by junk DNA is that in the bacterial chromosome, all of the nucleotides there are important. Some of them code for proteins, some of them are regulatory sequences like promoter regions or terminator regions, but they're all important. In eukaryotic DNA, it's really different. So all of our DNA, it's like, okay, so imagine this whole lab makes sense in the order that it is, right? If I took the staple out and then I took a bunch of random pages and threw it all up in there and then put it back together, this lab would all still be in there, but it wouldn't be in the same order. Or it might have a bunch of papers in between the different pages. Does that make sense?
Yeah.
That's kind of how our DNA is. It's like this picture here. So in this picture, all of these like purple, these gray, blue things make sense, but only if you put them together. Okay, right now there's a bunch of junk in between it. Okay, so this, all our DNA is like this. And why is it that way? It's a relative evolution. We'll talk about that in a different chapter. But what you need to know is in most eukaryotic DNA, we have in between all of our genes, there's a bunch of junk. Okay, the junk are called introns. just the word. The exons are the stuff that mean something. The reason it's given the word exon is because it means express. The exons are expressed. When I was in high school, I used to get, I don't ever know if I should say this or not out loud. I don't feel like you're always confused. But I used to always think exons exit. And I got it completely backwards. Exons don't exit. Exons are expressed.
No, I was thinking like exons. So I like.
Yeah, so however it works for you, but I used to always get it confused. But exons are expressed. Like my teacher never said exons are expressed. Like if I feel like the word heard, I would have made that confusion. But exons are expressed. So eukaryotes have a lot of junk that has to be taken care of before you can actually have a functional protein built. And we'll talk about how it's taken care of in a little bit. Okay, so that's kind of background stuff. Yeah.
But what happened if the introns were like?
I mean, they could, but so what we're going to have to do is splice our mRNA. If you don't slice it properly, then you'll make a polypeptide that's not going to function. That's what would happen. Okay, there's three. This is not in your book. You actually don't need to know this. If you want to pretend like you never heard it, fine. But just FYI, some people find this stuff interesting. There's three different polymerase enzymes in eukaryotes. Polymerase 1 makes ribosome RNA. Polymerase 2 is the one that we talk about most of the time that makes the messenger RNA. And polymerase 3 also does rRNA. Anyway, polymerase 2 is the one we talk about. So, like I said, the major difference, you do know this now, between bacteria and eukaryotes is the initiation of transcription. The initiation. Remember how I talked earlier also about regulation? Really important to regulate what genes are expressed versus what genes aren't. The initiation of transcription is the most important way to regulate gene expression. If you don't initiate transcription, you don't even start the expression process. So it's the most important step in regulating gene expression is the initiation of transcription. If you fail to do this, then you won't even read the genes and do anything. So how does it happen in eukaryotes? Well, think about, we have 46 chromosomes, and on each one of those chromosomes, there's literally thousands of genes. We have a ton of genes. So if we are like, okay, we need to express, gene 72B on chromosome 4. Okay, so your RNA polymerase will go in. Well, if RNA polymerase can bind to any random promoter region, what's going to prevent it from binding to, chromosome 2 instead and transcribing something over there? It has to be a little bit, well, really a lot more specific versus bacterial cells. There needs to be a lot more steps in place. It can't just bind any random promoter. It needs to bind only to a certain promoter. So in eukaryotes, there's a lot of other proteins that are involved before initiation can be complete. We call these proteins transcription factors. In this picture, they're the purple balls. So, at the beginning of promoter region in eukaryotes is a region called the Tata box. What it's called because the sequence on one of the strands is TA, 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 on the Tata box. On every eukaryotic gene, the beginning of the promoter region, there's a Tata box. So, when a gene needs to be transcribed, another gene somewhere else will produce a transcription factor that will then go and bind to this Tata box. That has to happen first. Once the first transcription factor has bound to the Tata box, additional transcription factors will come and bind in and around the promoter region. These other purple balls on this picture. Once they bind, then RNA polymerase can bind to the promoter region. But RNA polymerase will only bind if all these transcription factors are in place. If they're not there, it physically can't bind. So you might wonder, well, how does it help the RNA polymerase bind? The way they bind to the DNA extends it. It changes the shape in such a way that it makes it easier for the RNA polymerase to physically bind. So if they're not there, the RNA polymerase physically can't bind. Okay? Lots of them. So the first one is the one to the top of the box. Yeah. All right, once that happens, you have something called the transcription initiation complex. And Initiation is finished. Then elongation and termination are basically the same. Goes and reads the gene, builds the RNA transcript, BC here, good turn, falls off. Everything else is the same. Now so we can watch our video. Sore.
Transcription is the process of making RNA from a DNA template. Several key factors are involved in this process, including DNA, transcription factors, RNA polymerase, and ATP. Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is the Tata box. An enhancer region may also be involved. Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D, the largest of the general factors. A component of this factor, TBP, binds to the DNA using the Tata box to position TF2D near the transcription initiation site. Other transcription factors, including TF2A and TF2B, then attach. These complexes prepare the DNA for the successful binding of RNA polymerase. Once RNA polymerase is bound, other transcription factors complete the mature transcription complex. Now, energy must be added to the system for transcription to begin. This energy is provided by the reduction of ATP into ADP and PI. RNA polymerase then synthesizes an RNA template from the strand of DNA. Most factors are released after transcription begins. When the end of the transcription unit is reached, the RNA polymerase dissociates and the newly formed strand of RNA is released.
Okay. Did I help a little?
Yeah, I hope a lot of,
No. Oh, okay. All right, now, I'm going to skip ahead for a second and show you something. So, if you are a bacterial cell, the picture on the left, okay, there are no introns, so there's no junk. So as soon as you make an mRNA, it can be translated right away to produce a functional polypeptide. And most of the time, transcription and translation are coupled in bacterial cells. So here's the DNA, little line is the RNA, and here's RNA polymerase, and these purple things are the ribosomes. So as something is being transcribed, the mRNA is already being translated at the same time. They're called polyribosomes, those are lots of ribosomes. But anyway, we say that in bacterial cells, transcription and translation can be coupled. Because it doesn't matter because there's no introns. But this cannot happen in eukaryotes for a number of reasons. One, in eukaryotes, the DNA is in the nucleus. The RNA is in the nucleus, but the ribosomes are out here. So they physically can't get there. But also there's a lot of junk in this mRNA when it's first made, and it has to be taken care of and disposed of before it can finish and go be translated. So that's what we're going to talk about now, what happens with all that junk. Okay. So once we have what we call the primary transcript or the pre-mRNA, okay, so here's the DNA with the introns and exons. Here's the RNA with the introns and exons and the RNA. We have to take out all the introns. All the orange stuff has to be taken away and the green exon has to be put back together. That process is called RNA splicing, and it has to happen before the RNA can leave the nucleus. I'll get back to that in a second. The other things that have to happen before the RNA leaves the nucleus is the addition of something called a modified guanine cap. It's added to the 5' end of the RNA. Remember, the 5' end is where there's a phosphate group. Basically, guanine with 3 phosphates that's added there. That modified guanine cap has a couple of functions. The main function is it serves as sort of a flag for the small ribosomal subunit to bind to initiate translation when that needs to happen, because translation happens towards the 5' end of the RNA. It also can help prevent against degradation from enzymes in the cytosol, for a little while anyway. On the other end, the 3' end, something called a poly A tail is added. The poly A tail is a bunch of adenines, repeats, like 50 to 300 or so of them, added on that end. The poly A tail helps it to leave the nucleus. It assists it in leaving one of the nuclear pores somehow, but it also helps prevent against degradation from enzymes in the cytosol, at least for a little while. So those 2 things have to be added, and then splicing has to occur, and then we have what we call the mature mRNA transcript that's ready to leave the nucleus. So how does splicing happen? And I'm going to show you a video on this stuff. Splicing happens with these things called spliceosomes. That's how you spell it. You have all these words. There's a lot of, like, it's in the PowerPoint I sent you. That's why I'm skipping them. So splicing, like I said, takes the introns out and puts the exons together, okay, to produce this mature transcript. It's carried out by things called spliceosomes. What is a spliceosome? A spliceosome is a bunch of proteins associated with things called SNRPs. SNRPs, that's how you, SNRPs are small ribonuclear proteins.
Oh my God.
That's a SNRP. Good Lord.
That's
It's
a,
a start.
that's a whole
SNRP.
That's a whole
Yeah, it's really long. That's why we say SNRPs. SNRPs. So like I said, you have this. I'm just going to do this picture. So here's one SNRP. So an entire spliceosome is made up of a number of SNRPs, plus it will have a bunch of other proteins. So it's a really big purple ball, but in reality, it's a bunch of proteins all put together. Okay. SNRPs, notice the red in here is RNA, right? And then the protein is outside, so a small ribonuclear protein. So it's protein plus protein. On the SNRPs are very specific. The SNRPs will hydrogen bond complementary to the ends of the introns. It will sort of flank them. And when it does that, notice, then this has enzymatic capabilities. It will cleave the phosphodiester bond here and over here, cleave it, and then it will splice the exons back together, create a phosphodiester bond between the exons. So it physically will cut off the introns and put the exons back together. That's what the SNRPs do, these spliceosomes. Okay? And there we go. What happens to the intron that's been cut out? It will be degraded, and the nucleotides will be recycled and be used again. Yeah. Yes. It happens in the nucleus after transcription before the mRNA leaves the nucleus. Because we got to get rid of the junk. Yeah. Yeah. Good question. Yes. Yeah. Unless a random mutation happens.
Will it randomly like cut off part of the exon? Like, has it ever?
Some mistakes could happen. It's not
Okay.
supposed to happen, but if it does, then you wouldn't produce a functional mRNA probably. But it doesn't matter because more and more, they're
Okay, it'll
produced
just
a
keep
lot.
making itself again.
Yeah, a gene is transcribed hundreds of times.
So it could have a chance of not actually being true, but since
Yeah.
we make so much, it wouldn't affect us.
Yes.
Okay.
Okay, we already talked about ribozymes. Okay. All right, one of the reasons why RNA can function like this is that it can base pair with itself. And sometimes the RNA bases have functional groups, and they can also hydrogen bond with other stuff because they're single-stranded. Anyways, I'm going to show you a video. let's see, licensing.
I swear.
The following animation will describe the process of RNA splicing, an important step in creating the mRNA that is involved in protein synthesis via the process of translation. Key factors in this process include RNA possessing introns and exons, and the spliceosome. Here we see an RNA molecule with a single intron. Several signals exist within the introns that are used in the splicing process. From the 5' end of the intron, these are GU, the A branch site, a pyrimidine-rich region, and the 3' AG. The GU and AG sequences define the beginning and end of the intron. Splicing is mediated by the spliceosome, which consists of several protein RNA complexes. The first step involves two complexes that bind near the GU sequence. The RNA is then looped, and 3 other protein RNA complexes bind. This final complex then undergoes a conformational change. Introns are non-coding RNA sequences that must be removed before translation. The process of removing the intron is called splicing. The intron is then cleaved at the 5' GU sequence and forms a lariat at the A branch site. The 3' end of the intron is next cleaved at the AG sequence, and the 2 exons are ligated together. As the spliced mRNA is released from the spliceosome, the intron debranches and is then degraded.
You don't need to know all the specific details, I just wanted you to see it. All right. Another thing we need to mention is something called alternative splicing. Remember at the beginning I said one gene might code for multiple polypeptides? How does that happen? Because of alternative splicing. Now, there's some slides on it, but I'm just going to talk about it using this picture. So, for example, in some instances, in this case, this was treated as one intron, this was intron number 2, and we made this product. Well, in another case, what if the cell treated this intron as an exon and left it in there? You'd produce a totally different mRNA that will produce a totally different polypeptide. And that's what alternative splicing is. Sometimes introns can be treated like exons and left in the mRNA transcript to make a different polypeptide. Does that make sense? That's alternative splicing. Easy enough. It would, so there's a whole lot of other regulation on top of this. Like, these SNRPs have to be produced. And so if you don't make the SNRPs that will link this, then it won't be cut off. Does that make sense? How does all that decided? That's beyond the scope of this class. And I'm not sure that they know. But it would, so if you're not going to want to cut it out, the SNRPs wouldn't be produced to cut that out. So that's like how directly that would happen. But yeah, and a lot of these things, these SNRPs, all this stuff, are short-lived. They're not going to last very long, so that the regulation can remain tightly controlled. You know what I mean? All right, now we have to move on to translation. I told you we're going to be talking the whole class. All right. So DNA, transcription and translation, like I said, are coupled in bacterial cells, but are not coupled in eukaryotes. Excuse me. Translation, no matter what happens in the cytosol, it involves ribosomes, right? Ribosomes will read the messenger RNA and produce in a polypeptide chain. A sequence of amino acids all bond together with what type of bond? Wait. What bonds hold together amino acids? Peptide bonds. Esters, linkages are in fat. Sorry. A long time ago. That's that. Okay. I know. We have a long way to go. Here's our DNA sequence. Our mRNA sequence, the A was cut off. This would be our mRNA sequence, yes? It's complementary. You see that? How do we go from this to that? This is like 21 letters, but there's only like 7 codons. Yeah, codons. So there are 20 amino acids, but there's only 4 nucleotides. So how could 4 nucleotides code for 20 amino acids? The codons, right. The sequence matters, and there's sequences of 3. We call it a triplet code. Why does that make sense? If it was just 1, obviously we could only code for 4 amino acids. What if it was 2? How many could we code for? 16. 2 to the 4 is 16. So there's 4 letters, and if they were combinations of 2, you'd have 16 combinations, but this is 20, so that's not enough. So then we go to combinations of 3. How many combinations can you have? 64, which is way more than enough, right? So that's actually what we have. Huh? This is every organism on the planet. The genetic code is universal. Don't be scared. What is it? Yeah, you're wondering if the other people are wondering too. So we have 4 different letters, A, T, C, and G, right? In our DNA. If each letter coded for 1 amino acid, we can only code for 4 amino acids. We can't do that. If we had like combinations of 2, how many different combinations can you make with 2? If you have 4 letters, it's 2 to the 4, right? Is that it? Or 4 to the 2. 16. That's not enough. 4 to the 3. 64. Yes. Yeah.
So why is it only 20? Like, what? Is someone just not make anything?
Did you read the chapter? We'll talk about it. Hold on. No, they all make something. We'll talk about it. You learned this in Bio 1 as well. So, just some, I don't even know if your book talks about Nuremberg and Versailles. So these guys in the 60s, which really wasn't that long ago, like the 1960s, I mean, to you, that's probably a long time ago, but it's not. What are you doing, Eddie? Yeah, when you're on your phone during class, it really annoys me. I'm just calling you. I think phones are like the downfall of our society. Apples are good for you. I know.
You weren't here when we took that test.
You weren't here.
It's okay. They didn't know that what like the code was or anything like that, and they didn't know what codons coded for what amino acids or anything, and they basically determined the first codon amino acid match. They were doing a lot of research, and in one of their test tubes, they had ribosomes. They had all the stuff to undergo translation, and they synthesized artificially a messenger RNA molecule, and they made it only have uracil. Like that's all it had on it. They plopped it in there. There were all the amino acids, all the stuff. They didn't know what polypeptide was going to come out, but they made an mRNA with just uracil. They let it sit there for a while, heat it up a little bit, just like increased reaction, and then after a while, they made a polypeptide that was a bunch of phenylalanine. Like, whoa, the uracil combinations must code for phenylalanine. Because all we had on there was a bunch of U's. And then we had a bunch of phenylalanine. So they were the first ones to sort of connect this. Anyway, you can imagine the same thing. The mRNA with a bunch of A's on it. What do you get? What's the AA code for?
I don't get
I think.
Yeah.
Yeah. Alright, so they are, yay. Alright, let's move on. The triplet code, so that's the 3 nucleotides will code for 1 amino acid. Now, I want to use this diagram to talk about a couple things. The codons are mRNA codons. The codons are in the mRNA. Now, how is the mRNA made? It's used with the DNA. So the genetic code is really in the DNA, but the codons are the mRNA. It's important to understand that. Now, if the DNA is mutated, you're going to make the wrong codon. You make the right polypeptide, right? But the codons we talk about are the mRNA codons. Okay, so let's go to our codon chart. Here we go. This is called a codon chart. This is the genetic code. This is what you're gonna have. You're gonna have to know how to use this. The way you use it is to investigate what AAA codes for. First base, you find an A. Second base, another A. So A. Third base, look over here, A, lysine. Okay. You can use the circular one. I know. I like the circular one a lot better. Honestly, I didn't even know that existed until I started teaching. I was never exposed to it in high school or college. I was only ever exposed to this, but since I saw this, I was like, whoa, I like the circular one a lot better. On the AP exam, you'll be given the one on the board. Okay? You have to know how to use the one on the board. It's not hard. So,
I don't know, I think the one on the board is much easier.
whatever works for you. On the test, I will give you both, but on the AP exam, you only get the test.
All right.
The genetic code. That's what we're talking about. This is genetic code. There are 3 characteristics of the code you need to know. The first one is these 3 bullets right here. The first one is that the genetic code is what we call universal. Every organism on the planet follows the same genetic code. You can take a gene for a firefly, put it into a tobacco plant, and the plant will produce that like fluorescing color, just like a firefly. You can take a gene for a jellyfish that causes it to bioluminesce in the ocean, put it into a pig. During embryonic development, the pig will express that gene, produce that protein product, and it will fluoresce when you put it under black light. The pigs won't glow all the time. It only does it when it's illuminated with a black light, okay, which is why the pig back there looks kind of purpley. Anyway. Wait, what about the No, because the bacon doesn't come from the nose or the hood. It's only in certain tissues.
I wonder what the bacon on that was.
Right, it would have to be sodas or something. Now, they wouldn't sell this pig to be eaten anyway. This is a Y'all, if you've ever seen, we're going to talk about this stuff. We do a lot in biotechnology. The chapter, which is chapter 21. If you've ever heard of genetically modified organisms or GMOs, genetically modified organisms are genetically modified. A lot of peaches that are sold around the country that were grown by farmers were genetically modified to withstand frost and insect invasion and stuff, and a lot of them originated from Clemson Extension right over here. They've done a lot of the research at Clemson Extension here to develop a lot of these genetically modified peach plants. Right, some strawberries have an antifreeze gene in them, so they are protected from frost. So when farmers are growing them and it frosts early, they won't lose their entire crop. We will have a discussion about the advantages and disadvantages of genetically modified organisms. Some people think we shouldn't consume them at all. Some people think they're fine. Anyway, but that's my question. Oh, yeah. We'll talk about it when we do chapter 21.
Like the Carolina Reaper.
I mean, there's.
Oh, the pepper.
Well, that's not genetically modified. When I mean genetically modified, I mean you add a gene from another organism and put it in there. Okay, so listen, what they're doing with the reapers, that's not genetic modification by changing their axis, he's artificially selecting which ones to reproduce. So he's saying, I'm taking this plant and I am mating it with this plant. Does that make sense? So he's not adding a gene from another species. Yeah.
So
That video
I
was
mean,
off.
it's a ghost pepper.
Oh,
I mean, modifying happens all the time when we pick
Ghost
from
pepper
one.
arrow.
that's called artificial selection. I'm talking about evolution. Alright, the second, the genetic code being universal, this is one of the strongest pieces of evidence for what we call common descent. It is highly unlikely that all of the thousands upon thousands of species and organisms that live on this planet all evolve the same genetic code separately. Like, it's highly unlikely. Much more likely that evolved a few times way back when life evolved, and then we're all descendants of the same organism. Anyway, it's a very strong piece of evidence for what we call common descent.
Bacteria.
Over 3 billion years, yeah. It is impossible to imagine 200 years ago versus 200,000 years ago versus 2 million versus 2 billion. So, yeah, I mean, that's how long life has existed on the planet, about 3 billion. It's like, I can't imagine what it's like to live for 100 years.
Yeah, I can't imagine what it's like to live in the 50s.
Much less 100 million or a billion. Okay, the second characteristic of the code is not ambiguous. So when you were asking before, Colton, every single codon codes for something specific. So every combination will code for something. But what that also means is that sometimes there's overlap. Some codons code for the same thing. So the code is redundant. There's only 20 amino acids, but there are 64 combinations. So it has to be that some of these code for the same amino acid. If you look right here, all of these codons code for the same amino acid. They all code for leucine, right? All of these code for proline. All these are arginine. You get the drift. So the code is unambiguous and it's very specific, and it is redundant. A lot of codons code for the same amino acid. Okay. And it's a good thing to help protect against mutation. If in your codon you're supposed to have a C, let's say you're supposed to have a GAU, but instead you have a GAC. Does it matter? You're supposed to have this, but then you have this. It's the same amino acid. So if you have a mutation and like the effect is the third letter of your codon, you still might be okay. Because it might code for the same amino acid, and overall it has no effect on phenotype. Okay. Now there are, I'll take away my red. This codon is called the start codon, AUG. It's always the start codon. It codes for amino acid methionine. So the AUG is the start codon. There's only one start codon. Why do you think that's important? There's only one thing on here that codes for methionine. It's that one right there. It all starts at the same spot. You don't want multiple start codons. It wants to be very specific as to where translation begins. Okay, so there's only one start codon. There can be multiple stop codons. They're all up here. Stop codon does not code for an amino acid. It doesn't code for anything. It just ends translation. We'll talk about how that happens later. All right. So, how do they match to their amino acids? It's using something called transfer RNA. So let's look at what I just put up here. Here's a transfer RNA molecule. So the codon is AUG. Compare this to this sequence right there. Tell me what they are to each other. Complements to each other. How does a transfer RNA know? Like, I need to take methionine to this AUG. How does it know that? It doesn't know that. Part of the tRNA sequence has something on it called an anticodon. The anticodon is complementary to the codon. When you put them next to each other, they'll base pair together. It'll physically bond to it. And it just so happens to be carrying the right amino acids. They all carry the right amino acids. So how many different codons code for an amino acid? There's 64 combinations, 3 of them don't code for anything. 61. So how many tRNAs do you think there are? Around 61. Well, it can be a little less than that, but around 61. We'll talk about why it can be less than a little later. It's called wobble. Did you read about wobble? Yeah. Okay. But in general, there are like 61 or just a few less different tRNA molecules. The tRNA structure, let me just show you the picture. So this is like a structure of tRNA. So it's an RNA molecule. And one side of it is a specific sequence that is the anticodon. It is the complement of the codon. It is part of the tRNA molecule, yeah. So this is tRNA. tRNA is actually it's an RNA molecule. It base pairs together and it forms this like clover leaf shape that is actually on top of each other, so space filling looks like that. Then at the very bottom is the anticodon. Like I said, that little base pair complementary with a codon on mRNA. At the other end, the top over here, is where the amino acid will bind to. That's how it carries it. And on the sides here, the reason why they're different colors is because when the tRNA, I'm going to flip forward for a minute. When the tRNA gets inside of the ribosome and these sites, it actually physically will base pair to the ribosomal RNA in the ribosome. Where does it base pair? It's based on these sites right here. But actually physically it's held in the ribosome. So that's tRNA, codes for RNA. Like I said, there's 61 different codons that will code for the different amino acids, and there's around 61 different tRNAs. A few less because of something called wobble we'll talk about later.
Is there any way, like, the amino acid could be wrong on the tRNA, or is it always going to be?
Should always be right, because there's these enzymes. There's a specific enzyme that will add the amino acid to the tRNA. And if there's like 59 different tRNAs in one cell, there are 59 of different of these enzymes. Each one of these enzymes is specific to a tRNA and a specific amino acid that will bind to it. Yeah. tRNAs are reusable. Okay, it's sort of like your car. You can drive it to school in the morning, you get back in it and drive yourself back home. They're reusable. tRNAs are reusable. It will take a amino acid and drop it off, and it can go to get another one. The enzymes that catalyze the addition of the amino acids are these things called amino acyl tRNA synthase.
Oh my God.
That big one. Right here. That's this enzyme.
No, it's still started.
Yeah. So anyways, it takes energy to do that. You just need to know that they exist. These are in the cytosol, so whenever tRNA drops off an amino acid, it'll just go right back to one of these and get another one. A lot of work to teach this. It's good y'all are second. Well, I like you, and so I'm not as demotivated as I actually want to be right now. Not that I don't like the other class, but I'm saying, like, y'all are fun, and, like, I think the other class just have to sleep in the morning. But I'm, like, fresh, so, like, I'm okay to teach, but now I'm just, like, tired of saying the same thing again. But since y'all are, I don't know, it just makes it works. It works. All right. Right, a ribosome. Ribosomes have two subunits, a large and a small. So the large subunit has three different physical sites inside of it, the E, the P, and the A site. I'm going to start with the A site. The A site would be like the first site that a new tRNA will come into. A stands for amino acyl tRNA binding site. Go figure. Where that tRNA binds to. The P site is the site in the middle, and that's the peptidyl tRNA binding site. This is where the amino acid, you look at the picture down here, that has the growing chain of amino acids bound to it is in this site here. E stands for exit. So once a tRNA has released its amino acid, it leaves from the exit site to go get another amino acid. So those are the three sites in there. Ribosomes are made up of RNA. There's ribosomal RNA and proteins in there. The ribosomal RNA has a catalytic function to help catalyze the peptide bond formation to make this amino acid, and also the breaking of the bonds from the tRNA in that first site, or in the second site, you'll see. The RNA also in the ribosome will help bind to the tRNA to keep them stable in there, especially while they're in the A and the P site. The E site, if you notice in this picture right here, is a little bit shorter than these two sites. You see that? When the tRNA actually enters into the E site, because it's a little different shape, it doesn't stay there very well and it's kicked out. So that's why it exits from that site. Okay. There are 3 stages to translation: initiation, elongation, and termination. The names are the same as transcription, very different stuff happens. So I'm going to talk about all of it just using pictures, because I can't say words, but all this is in your notes, right? So this is initiation. So first stage of translation, this is the same in bacteria and eukaryotes. It's all the same, okay? Translation, initiation. The first thing that happens is that the 5' end, remember, of the mRNA is where translation needs to begin, where that modified guanine cap is located. So notice how we're starting at this 5' end. You see that in the picture? Yep. Okay. So one of the first things that happens, and is the binding of the small subunit and the addition of that first tRNA. It kind of all happens at the same time. There are other proteins involved called translation initiation factors that are not shown in these pictures, because it would make it really confusing. Okay, just know this doesn't happen just by itself. There's proteins that assist. So the small subunit will come and bind towards this 5' end, and it'll associate itself right around where the start codon is. Where the start codon, this first tRNA will bind complementary to it. Okay. Now, if the tRNA comes around and it's not complementary, it'll just bounce off, like it won't bind. But if one comes and happens to be complementary, it'll hydrogen bond there and stay there. It always carries methionine, this first tRNA. It's called the initiator tRNA. So once this happens, we have a small subunit, the first tRNA on the start codon. Notice the start codon is not the beginning of the mRNA. What's over here? We have our modified guanine cap and even some more regulatory sequence. Okay, so it won't be the beginning of it. It'll usually be a little bit downstream. Then, using the energy of DTP and some other proteins that help, the large subunit will basically come plop itself on top of it. So this is not initiator tRNA. This large subunit will come and like stick itself on top of it, like that. Okay. In such a way that that tRNA is in the P site. Once you do all that, that's called the translation initiation complex. And you're ready to translate. Questions about that? Some of y'all look entirely bored. Later the class, maybe I'm going to head nod. And funny. Okay.
Yeah.
Next, I'm going to go through all this. The next stage is called elongation. You orient yourself to this picture. Notice how we've been elongating for a little bit. If you go back here, there's only one amino acid on this side. If you go here, notice there's 4 now. We've already done a few rounds of this already, okay? So the amino acid chain that's growing is bound to the tRNA in this P site. So what happens next is that the A site, notice, is empty. The tRNA will pop in here, okay? If it happens to be complementary, it'll hydrogen bond. If not, it'll pop right back out, okay? But if it is, it'll hydrogen bond here. And also the sides of it will bond to the rRNA. Okay, here we go. So it's in there. The next thing that happens is that using the energy of DTP and the ribosomal RNA catalytic ability, this growing polypeptide chain will break off of this tRNA and will bond to the amino acid on the tRNA and the A site. So notice the difference between this picture and this picture. See that? The growing amino acid chain breaks off of the tRNA in the P site and will form a peptide bond to the amino acids on the tRNA in the A site. That red line is that peptide bond that we just formed using the energy of DTP. And the ribosome RNA catalytic ability helps catalyze all this to go on. Great, so we just made our polypeptide chain 1 more amino acid longer. Now, what needs to happen? This tRNA is empty. We need to go grab another one, and we need to go read the next codon. So notice the difference between this picture and this picture. What happens is that this whole ribosome is what we call translocate. It translocates itself the distance of 1 codon to the right. So it's like the mRNA moves the distance of 1 codon to the left. Okay? Now these tRNAs stay bound to their codon. They're stuck there. It's like the stuff moves around it. Okay? So if you're on like a little ride, okay, and the ride moves, like you're gonna move with the ride, but like it's a fair, kind of little ride, you know, and like it's moving and like the stuff's moving around you, but you're still in the same spot because you're attached to the chair. What's happening here? These are attached. So this whole thing moves around it, but it stays attached to where it's attached. Makes sense? It moves the distance of one codon. Okay? Such that this tRNA that was in the P site is now put in the E site. When it gets there, it doesn't fit very well anymore, it's kicked out. The tRNA that was in the A site that has the growing polypeptide chain is now put in the P site. And the A site is empty. And we repeat over and over and over and over and over again until a stop codon is reached. Once the stop, this is termination now. Once the stop codon is reached, something called a release factor comes in. A release factor is not carrying any amino acids or anything. They don't, the stop codons don't code for anything. But a release factor comes in. A water molecule is used to break this bond here to release the polypeptide. The tRNA is released, and then everything falls apart. It uses some energy to cause everything to deattach to itself. And that's the end of translation. Let's watch some videos. Go. Yeah, have fun. Have fun. Good luck.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when a small subunit of the ribosome attaches to the cap and leads to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and more tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Hi. I have one more.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now used in...
I'll see y'all tomorrow.